right, hello everyone, welcome back. This is week 3 of our little science facts course. Um, Last time we talked about characteristics of life. We talked about some cellular structures, basic ones. We talked about the virus structures. And last week we also talked about mitosis, which is the basic way for eukaryotic cells, which are like our human cells, how they divide. Um, and then today we're gonna get into viruses, which should be really fun because of what's going on right now. So, first thing I'm gonna introduce is just to give you some context as to what viruses are compared to to our normal cells, and how amazing they are in their diversity and in their numbers as well. um, it is kind of scary, but at the same time, not all viruses are bad. um, we also use them um, for transferring DNA and stuff, so they're very useful in the science community as well, and they help us develop. So you always need some kind of rival to help you develop. Cells need that as well. So this is a picture showing just some cells, and then you can see those big dots are just normal cells, and then you can see all of these little dots everywhere. These are viruses. So this is somewhere in the ocean. This is just uh, a little screenshot of just some cellular structures, and then you can see how the viruses are way, way, way more than the cells are, and they're way smaller. So um, that's just some little context as to how different viruses are from cells. And then, as we said before last time, we said viruses are not alive technically by the standards that the science community set. They're just a group that we have. We are not very. We're not very. We cannot easily define because they're just so different, and within there's so many different types of viruses and they all do different things, so we're not really sure how to um, how to categorize them as well as we do for like human cells or for bacteria. So they're also, we said, really small. So if you want to compare, these are pictures of viruses, virus particles over here, these little dots, and I'm guessing that's in a cell. They're breaking it apart. This is a bacterial cell, and this is a eukaryotic cell, one of the smallest, which is a red blood cell. So this is what makes your blood. These are the cells that make your blood. So if you want to compare, you look down here, you see this is how big viruses are compared to some of the smallest cells, which are bacterial cells. See how small they are in comparison? And this is how small a bacterial cell is compared to some of the smallest cells that we humans have, which is a red blood cell. It's not really that clear, but you can see how big they are. So this is how small a virus is, and this is how a human cell is compared to it. See the vast difference. And then we can only mainly see them under microscopes, electron microscopes, which are different than normal microscopes because they can see very, very, very small particles down to the atoms. It's very amazing. Also, just another size scale to give you. So a virus compared to just a normal balloon is 10 million times bigger than a virus. The same way that the whole planet of Earth is 10 million times bigger than a balloon. So compare a balloon to the size of Earth. That's how small a virus is compared to just one small balloon. So viruses are really, really small, very almost impossible to detect, but at the same time, because of their numbers and their diversity, that they are able to actually have an impact on other living things. Some of them are large, but overall they're very small. So these are just little tiny snippets of some of the large viruses. This is the Pandora virus, this is the Pythora virus, and Some of them are a little big enough to detect near other normal cells. So, remember last time we said viruses are so diverse and they're so different from each other that they kind of almost have nothing in common except for two things. So, two things that the viruses have in common. They have the nucleic acid genome, which we said is like the DNA. There's different types of genetic information. So we have DNA, RNA. So viruses have that. So they have genetic information, which is what makes them them. And then they have something coding that information, kind of protecting it, like a little, like a little capsid. So we call that the protein capsid. So these are the only two things that viruses have in common with each other. Everything else is they're so different from each other that it's impossible to, put them in categories or, give them names. So so we said the DNA in viruses is so like right here, for example, if you look at the coronavirus, you see. So these little over here, these little coils, that's the DNA. So last time we said the DNA, that's the information that makes you you. So the cells use that to make proteins, and these proteins make everything that we know. So that is the information that basically carries your identity. So viruses have different. So we humans have DNA, right? And then we make it into RNA, and then we make it into protein. Viruses carry their genetic information in different ways. So it can be DNA, it can be RNA, it can be different shapes in which it can be doubled, and it can be single, it can be in a circle shape, it can just be in a line. So they're very diverse, even in the way that they carry their genetic information. So these are just some examples of the type of genetic information. You don't need to know that for now, that's too complicated. And then these are just some of the shapes that the bacteria are in. So over here you can see this is called the tobacco mosaic virus. It's like in this linear shape. And then the adenovirus is kind of like diamond shaped. You can see it right here. It's in this shape. And then influenza virus is circular, kind of like the coronavirus. It's also circular, and it has an envelope. It doesn't really show that clearly here, but that's what the influenza virus looks like. And then you have the bacteriophage. So these are some of the basic viruses that colonize bacteria. So viruses do not only invade our cells, like human cells, but they also invade bacterial cells as well. So this is the bacteriophage. It looks really cool. Kind of looks like an alien. This is where it houses its genetic information, and then it gets into a cell using these little spider things. This is how it attaches to a cell and then takes its information down into the cell. So these are just, again, talking about the basic ways. So if the virus is shaped like this, it keeps its DNA coiled inside like that. And if it's shaped like this, keeps in its DNA like that. So you can kind of see where the DNA will be housed, where the genetic information. I shouldn't keep saying DNA, because it's not just DNA. It can be DNA or RNA, but this is how it houses its genetic information. So we're gonna get into the basic steps of viral reproduction. Again, viruses are so diverse that they can do a lot of things differently. Some of them have different kinds of proteins in them that maybe make the process easier. But these are just some very basic steps into how a virus gets into a cell, invades it, and gets out of it with many more viruses than before. So the first step is attachment. So it has to have a way to attach itself to the cell. Remember last time when we talked about cells, we said that the outer layer of every cell, the plasma membrane, it doesn't just let anything in and out. It has to have some kind of recognition, like some kind of like key, some kind of password. So these proteins on the outside of other cells, they recognize each other, and then they might let each other in. So it's not that easy. And then we have entry. So this is just how it enters the cell. Once you're in the cell, you kind of also need to, be more uh, be more low key about it. So you can't just enter the cell and do whatever you want. There's also things in the cell that might attack you if you're an invader. So if you when you enter the cell, you also need some kind of mechanism to help protect you from being degraded or destroyed. Um, and then the very important step is once viruses enter cells, they don't want to do anything other than just make more genetic information. So they want to just make copies of themselves. That's all they want to do. So once they enter the cell, they just have to find a way to make more of their genetic information, whether it's DNA or RNA or whatever. They need to make that information into, so they divide it and then they make proteins out of it and then they leave the cell again. So then once they divide over here, they assemble their parts together, so they make the cell do the work for them, which it divides the DNA for them or the RNA, and then some of it gets made into proteins, some of it just stays in those over here in that shape. So over here it divides this part, which is the genetic information, and from this genetic information it makes proteins like these over here, which is like the capsid and these outer proteins over here. So it makes everything from that cell using this little coil of genetic information. So it makes everything, assembles it all together into a virus again, and then it exits the cell. So there's also different types of exits that it can make, in which some of them hurt the cell, some of them don't hurt the cell. So depending on how it does it. So this is a basic example. This is called the lytic bacteriophage. So lytic means that that it will destroy the cell when it gets out of it, so it doesn't just get out nicely the way it entered. It gets out by like exploding from the cell, and this way the cell actually dies because you're literally just exploding the cell. So this looks a little complicated, but it really isn't. So the virus this is an example of a bacteriophage, which is those little alien-looking viruses that live in bacteria. So this is a bacterial cell, and this is a virus. So it comes in, attaches using those little spider proteins, and then you can see the DNA get out of it, like kind of squiggle out. And then this is a way where the virus does not, so this is the bacteria cell's own DNA, and this is the viral cell's own DNA, right? So in here, it doesn't need to go into that DNA, the virus DNA. It just uses some of the cell's proteins to make more of this. It doesn't care about this, right? So this is the viral DNA. It makes copies of it and it translates it into mRNA. And then this mRNA gets made into proteins. So you can see these are the virus parts over here, right? So we just went from one of these, so one virus DNA, into many virus DNAs, and then some of those it took and it made into proteins over here. So now you have all the parts of the virus that you need. You have the DNA, and then you have the proteins, right? So now you just need to put them together. You need to assemble them so they can get out together as a whole virus, right? So you started with one, you make copies of everything, and now you got them all assembled together. So now you just need to get out. So in here, they just burst out of the cell, leaving the cell destroyed. So you started with one, now you have tens, sometimes hundreds or thousands of them from one cell. So this hurts the cell in that cells use up a lot of energy when they divide stuff, when they make stuff into proteins. So you're using up the cell's own like energy, you're using up the cell's materials, and you're making more of yourself. And then in the process, you also, when you get out, you destroy the cell. So this is how other organisms are affected by viruses. So they exhaust their materials and their energy, and then they destroy them, right? But not all viruses destroy the cells in the process, right? Some of them do it more discreetly, right? So this is a lysogenic bacteriophage. So lysogenic bacteriophages is um, different in that they don't just come in, make copies of themselves, and then get out really quickly and destroy the cell. Some of them like to stick around a bit longer. You know, when the cell is doing fine, when the environment is okay. They can just what they do is they just stay in the cell for a really long time, and they when the cell divides, it divides with it. So this way, this one over here, it's very quick because it doesn't mix with the cell's DNA and then makes more of itself. It just makes more of itself, sorry. It doesn't mix with the cell's DNA, it just makes more of itself really quickly and then gets out really quickly. This one, it, it enters the same way and then it mixes its genetic information, its DNA, with the cell's DNA. So you can see it over here, this part. It's mixed with the cell's DNA. And then I remember last time we talked about mitosis, we said with mitosis over here, when the cell divides, right, it divides its DNA first, right? Divides its DNA first, and then the cell divides. And here, what the virus does is it just gets into that DNA so that when the cell DNA divides, the virus DNA is also dividing with it. So it does this in a way where it doesn't really hurt the cell. It's just hanging around in the cell. This generally happens when the environment overall is good, so the bacteria doesn't really need to just come in, do its thing, and then get out really quickly. It's just chilling. So this way, when the cell divides, it divides with it. But then when things start to get shaky, like around, I don't know, the environment is not so good around the cell is not doing so well, or the cell is getting old, what it does is it takes out its DNA from the cell and then divides and then assembles its part and leaves. So when the environment is not so good anymore, it's not so perfect for the virus. It takes out its DNA the same way it put it in and then it leaves. Right. So then these are the steps combined. So this is the virus coming in. So then we said so this virus doesn't really have an envelope, it just goes in, it puts its DNA in. Some viruses, like the coronavirus, for example, or the HIV virus, they have this envelope thing so that when it goes in, so over here it uses those proteins we said to kind of like a password into the cell, and then it tricks the cell into letting it in, right? So once it goes in, it has this little extra envelope, right? That lets it survive in the outer. So this is in the cell, but then this is the nucleus where the DNA is. So it lets it kind of survive around here so that when it goes into, so that it has a chance to actually get into the DNA, right? So viruses that need to integrate themselves into the cell DNA, like over here, viruses that need to mix in the cell DNA, this is a bacteria. Remember we said bacteria don't have the DNA is just hanging around in the cell. It's not enclosed in a capsule. But then like a normal cell, like our cells, the DNA is closed into an extra, like think of it as like a different room within the cell that keeps the DNA in it, right? So the virus needs to get into that room. So got into the cell, but now needs to get into that room. So what it does is it has two extra layers, right? It has the envelope and then it has the capsid. So the envelope, the first layer, it lets it into the cell, right? Now it needs to get into that room. So that second layer lets it go into that room without harming the actual DNA of the virus, right? It lets it get into the cell and then get into the DNA, get into that room, the nucleus, and then lets it get into the DNA and then do its thing. It can hang around for a long time, or it can, and then eventually it will get out the same way, make copies of itself, and leave the cell. So this is the basic way that viruses enter and get out. I do not, I think I got into enough detail for today, but next time we'll talk about different viruses, and then maybe we'll get into some more detail. But this is just some basic ways that viruses enter cells and they make more of themselves and they get out. That's all what viruses do. They don't really try to grow or develop or anything. They just make more of themselves. And in that process, some of our cells get hurt. So that's what happens during a viral invasion.